Hello and welcome, dear students. Today we are going to discuss about the topic population and disease association studies. The main objectives of today's lecture are to know the basics of association studies, to know the history of association studies, to know about technical aspects of association studies, to understand the allelic spectra of common diseases, to discuss about the challenge in association studies, and finally to discuss about the risk assessment in association studies. Dear students, let's start with the introduction first. The population association studies are done to identify patterns of polymorphism that vary systematically between individuals with different disease states and could therefore represent the effects of risk enhancing or protective alleles. There are two basic types of study designs used for genetic association studies: the case control study and the family based study. Both types rely on the concept of linkage disequilibrium between the alleles at a genotyped marker and the disease allele. The development of common disease results from complex interactions between numerous environmental factors and alleles of many genes. Identifying the alleles that affect the risk of developing disease will help in understanding disease etiology and subclassification. Over the past 30 years, genetic studies of multifactorial human diseases have identified more than 50 genes and their allelic variants that can be considered irrefutable or true positives. However, there are probably hundreds of susceptibility loci or positions that increase the risk for each common disease. Large-scale association studies hold substantial promise for unraveling the genetic basis of common human disease. A well-known problem with such studies is the presence of undetected population structure, which can lead to both false positive results and failures to detect genuine associations. The consequences of population structure on association outcomes increase markedly with sample size. For the size of study needed to detect typical genetic effects in common disease, even the modest levels of population structure within population groups cannot safely be ignored. Recent advances in genotypic technologies and increase in genetic marker availability have paved the way for association studies on genomic scales. A potential problem for every population-based association study is the presence of undetected population structure that can mimic the signal of association and lead to more false positives or to missed real effects. These concerns have influenced the design, interpretation, and funding of association studies during the past decade. Still, levels of population structure in many ethnic groups are typically small, and despite concerns, there is an increasing sense that the problem is not serious if association studies avoid gross levels of population structure. Future association studies will genotype many markers and evaluate many individuals, owing to the realization that case control studies' power to detect realistic effect sizes will typically require thousands of individuals. This concern raises two general questions. One, how much underlying structure is there in various human populations, and when might this pose problems for large-scale association studies? And number two, how accurate and efficient are available methods for correcting for population structure in case control studies? Dear students, human geneticists seek to understand the inherited basis of human biology and disease, aiming either to gain insight that could eventually improve treatment or to produce useful diagnostic or predictive tests. As recently as 2004, few genetic variants were known to reproducibly influence common polygenic disease, including cancer, coronary artery disease, and diabetes, or quantitative phenotypes, including lipid levels and blood pressure. This relative ignorance limited potential insights into the pathophysiology of common diseases. The completion of the human genome sequence in 2005 and the provision of an initial catalog of human genetic variation and a haplotype map known as the HapMap, together with rapid improvements in genotyping technology and analysis, have permitted association studies to be undertaken in a large number of samples. The association studies, in which hundreds of thousands of single nucleotide polymorphisms or SNPs are tested for association with a disease in hundreds or thousands of persons, have revolutionized the search for genetic influences on complex traits. Such conditions, in contrast with single gene disorders, are caused by many genetic and environmental factors working together, each having a relatively small effect and few, if any, being absolutely required for disease to occur. Although complex conditions have been referred to as the geneticist's nightmare in the past 5 years, genome-wide association studies have identified SNPs implicating hundreds of robustly replicated loci, that is, specific genomic locations for common traits. Despite their value in locating the vicinity of genomic variants that may be causing disease, few of the SNPs identified in association studies have clear functional implications that are relevant to mechanisms of the disease. Narrowing an implicated locus or a position to a single variant that directly causes susceptibility to the disease by disrupting the expression or function of a protein has proved elusive to date. This will be a key step in improving the understanding of the mechanisms of disease and in designing effective strategies for risk assessment and treatment. Dear students, let's know some key points about the disease association studies. Recent improvements in genotyping technology and in the knowledge of human genetic variation have made it possible to carry out genome-wide genetic association studies to identify susceptibility genes for common diseases. The allelic spectrum of complex diseases, that is the range of the frequencies and effect size of susceptibility loci, will influence the success of association studies. Large sample sizes will be required to search even the most accessible end of this spectrum. The existence of extensive regions of linkage disequilibrium in the human genome will greatly reduce the cost of genotyping in genome-wide association studies, as this allows the use of tag SNPs that provide information on a number of other SNPs that are not directly genotyped. However, as much as 30% of the common variants in the genome remains unknown, and the latest high throughput technologies convert only about 50% of SNPs into robust assays. In addition, extensive resequencing and genotyping will have to be carried out to ensure that SNPs in regions of low linkage disequilibrium are surveyed comprehensively. Other factors that need to be taken into account in the design and execution of initial association studies to avoid loss of statistical power include possible selection bias, population structure, and misclassification errors. Dear students, let's know about the history of association studies. Nearly 600 genome-wide association studies covering 150 distinct diseases and traits have been published, with nearly 800 SNP trait association reported as significant. Such associations are consistent with the common disease common variant hypothesis. This posits that genetic influences on susceptibility to common diseases are attributable to a limited number of variants present in more than 1% to 5% of the population. The common disease, common variant hypothesis is exemplified by susceptibility to age-related macular degeneration. 5 major variants are associated with age-related macular degeneration, and each is associated with a risk of disease that is 2 to 3 times the risk of a person without one of the variants. 2 of these variants are found in the complement factor, that is HCFH gene. Taken together, these 5 variants more than double the risk of age-related macular degeneration in the siblings of affected persons. This accounts for roughly half the estimated total risk for siblings and suggests that the complement-mediated inflammation pathway is central to pathogenesis. The discovery that inflammation plays a role in age-related macular degeneration and is proving to be a suitable target for therapeutic intervention in animal models. This demonstrates the power of the genome-wide association study to implicate previously unsuspected pathways in the cause and pathogenesis of disease, leading to the development of new therapies. The genome-wide association study has yielded more than 30 variants related to Crohn's disease. 3 of these variants found in the genes nod to IL-23R and LRRK2 are common. All but one have risk allele frequency of more than 9% in the population studies and are associated with an increase in risk by a factor of 1.5 to 4. A similar pattern of a few variants having large effects but most having small effects has emerged for type 1 diabetes, with more than 40 variants identified to date. Other common conditions have not been as amenable to investigation of genome-wide associations. An early example was schizophrenia. 5 association studies failed to find any variants reaching genome-wide significance. A 6th study implicated rare structural variants that disrupt neurodevelopmental pathways, raising questions about the role of structural variants in neuropsychiatric disorders. Subsequent large studies investigating the risk of schizophrenia have implicated several variants, both structural variants and SNPs in the region of the major histocompatibility complex, or MHC. Generally, associations between SNPs and traits or characters tend to be of modest effect size, with a median odds ratio per copy of the risk allele of 1.33. Several variants carry odds ratio above 3, including some exceeding 12. These are of particular interest since it seems likely that there would have been evolutionary pressure against their selection unless they provide some survival benefits in earlier periods or different environments. This is not to imply that smaller odds ratio are unimportant. The genes PPARG and KCNJ11 associated with type 2 diabetes and IL-1-2B associated with psoriasis encode proteins that are targets for thiazolidinodiones, sulfonylureas, and NTP40 antibodies respectively, but all have odd ratios less than 1.45. Such variants may shed light on the pathophysiology of their associated traits and reveal new therapeutic targets. Only 12% of SNPs associated with traits are located in or occur in tight linkage disequilibrium with protein coding regions of genes. Although SNPs in protein coding regions are heavily overrepresented on genotyping arrays, approximately 40% of trait-associated SNPs fall in intergenic regions, and another 40% are located in non-coding introns. These 2 findings have sharpened the focus on the potential roles of intronic and particularly intergenic regions in regulating gene expression. The potential roles of the complement system in age-related macular degeneration, a disease previously thought to be primarily degenerative in origin, or of autophagy in inflammatory bowel disease, for example, were not widely suspected until these systems were implicated through association studies. Similarly, association studies have identified loci that are shared by conditions previously thought to be unrelated. The possibility of common etiologic pathways in such disparate conditions or traits are type 2 diabetes and invasive melanoma, Crohn's disease and Parkinson's disease, or prostate cancer and height raise intriguing questions about the pathophysiology of these seemingly unrelated conditions. Also, about the potential for using drugs that are effective in the treatment of one condition for the treatment of other. Dear students, let's now discuss about the technical aspects of association studies. Association studies build directly on recent efforts to map the patterns of inheritance for the most common form of genomic variation, the SNP. An estimated 10 million common SNPs with a minor allele frequency of at least 5% are transmitted across generations in blocks. This allows a few particular SNPs to capture the great majority of SNP variation within each block. Rapid advances in technology and quality control now permit affordable, reliable genotyping of up to 1 million SNPs in a single scan of a person's DNA. Scanning can be used in various study designs, including case control studies, cohort studies, and clinical trials, as long as it is recognized that the known strengths and weaknesses of these designs are pertinent to the use of scanning. A complication of association studies is the enormous number of tests of association required, at least one per SNP or single nucleotide polymorphism. The threshold of statistical significance are stringent, making it necessary to work with very large samples. One frequently used approach to managing size is the TIRAD design, in which a subset of SNPs found to be significant in the genome-wide association study, sometimes called the discovery set, is genotyped in a second tier, a replication set. This yields a smaller subset of significantly associated SNPs that are then tested in a third tier, a second replication set, and so on. This process helps to identify false positive associations. Carrying forward, a large number of SNPs identified through a genome-wide association study into a test of replication also minimizes false negative results while raising the bar for the establishment of true positive results. The pooling of results obtained in association studies under the auspices of large consortia is often required for the detection of variants with small effects on the risk of disease. Such pooled studies, like all genetic association studies, must be examined and controlled for differences in allele frequency between groups that can lead to spurious false positive associations. The most reliable evidence of a true genetic association, short of defining the causal variant functionally, is replication of the association, especially if it appears in multiple populations. Dear students, let's talk about the allelic spectrum of common diseases. The allelic spectrum or architecture of a disease refers to the number of disease variants that exist, their allelic frequencies, and the risk that they confer. Numerous sources from both theoretical models and practical experiments have provided insights into the allelic architecture of common diseases, demonstrating the multiplicity of loci that are involved and their range of effects. Regardless of the exact shape of the spectra, which will differ between disease, and the allele frequencies of variants that predispose to disease and the strength of their phenotypic effects indicate the potential statistical power of genetic association studies, and therefore their likelihood of success and the cost per true positive result. It should be noted that other factors also affect statistical power. For example, confounding factors such as population structure and geography, misclassification errors, and selection biases. Dear students, let's now discuss about the challenge in association studies. Trait-associated SNPs may point the way towards functional genetic variants but are unlikely themselves to be the causative variants, at least given the current understanding of genomic function and regulation. At first step in narrowing a genome-wide association signal to potentially causative variant is to type all the known SNPs in the haplotype block represented by the tag SNP, a process known as fine mapping. This is to determine whether one of these SNPs has a stronger association than that tag SNP or an established functional effect. Although this approach has shown promise in identifying casual variants, its yield has been limited. Extensive sequencing of an associated region may identify additional previously unknown rare variants, frequency less than 1%, with a possible biological role. Given the lack of good representation of SNPs with a prevalence of less than 5% in current genome-wide association arrays, a comprehensive catalog of SNPs with a prevalence of 1 to 5% is being generated by the 1000 Genomes Project for potential inclusion in fine mapping efforts. Annotation catalogs, that is the maps of functions of variants such as those related to transcription factor binding, that is promoting gene expression, or to RNA interference, that is gene silencing, are currently in development and should facilitate the identification of functional variants underlying association signals. The small proportion of heritability and risk of disease typically explained by genome-wide association findings present a challenge as how to identify the variants that confer the outstanding risk, the risk that has not been accounted for. Large genome-wide association studies that identify more variants are likely to identify variants with even smaller effect sizes. The importance of structural variation, including copy number variants, inversions, and translocations, is an active area of investigation. Several structural variants underlie association signals for autism, schizophrenia, Crohn's disease, and obesity. The older populations which have undergone more mutations and a greater number of recombination events have greater degrees of genetic variation and shorter stretches of linkage disequilibrium, allowing for better localization of association signals. Dear students, let's now understand the risk assessment in association studies. The potential for variants identified in association studies to predict the risk of complex disease has been anticipated since the publication of the first reports, but this application is problematic. For the prediction of complex diseases, genotypes at multiple SNPs are often combined into scores calculated according to the number of risk alleles carried, which is the approach that is used in predicting the risk of cardiovascular diseases. On the basis of 9 SNPs associated with cholesterol levels, this score is believed to be strongly associated with this risk of cardiovascular disease even after adjustment for standard risk factors, including family history. It has also been argued that the use of dense genotyping information from tens of thousands of SNPs with only nominal associations with disease may improve the accuracy of phenotypic prediction. Care is needed in evaluating genetic predictive models since they are often specific to the population in which they were developed, and their value can vary with genotypic frequencies, effect sizes, and disease incidence. Possible clinical uses of predictive scores, for example, in deciding which patients should be screened more intensively for breast cancer with the use of mammography, or for statin induced myopathy with the use of muscle enzyme assays. This will require rigorous, preferably prospective evaluation before being accepted into clinical practice. Genome-wide scans permit screening for many conditions at once. A binomial probabilities were applied to 40 independent diseases. For example, roughly 90% of the population would be placed in the top 5% of those at genetic risk for at least one of the diseases. 33% would be in the top 1% and 4% would be in the top 0.1%. So expanding such screening to 120 diseases would nearly triple the proportion in the top 0.001% at risk and identify 1.2% at the top 0.01% levels that could justify population-based screening if appropriate interventions were available. The ability to assess risk for 120 conditions at the same time also raises the concern that predictive models will yield conflicting recommendations if implemented that would reduce a person's risk for development of one condition and worsen the risk for development of another. Such considerations are timely and important since several commercial ventures are marketing association-based screening directly to consumers. This testing can often be obtained without a physician's intercession and has been promoted for medical, genealogic, and even recreational purposes. Few factors associated with differences in risk across a population will separate affected and unaffected groups widely enough to be useful for individual prediction. Adequate communication of disease risk is a topic that has challenged generations of physicians and patients, and the perception of risk is more often influenced by emotion than by science. Genome-based risk information may not improve communication of risk, but its uniquely individual nature may be personally motivating and could be explored with respect to the promotion of salutary behaviors. Dear students, let's now conclude the lecture. Association studies have proved successful in identifying genetic association with complex traits. This reasonably unbiased approach to surveying the genome has opened doors to potential treatments by revealing the unexpected involvement of certain functional and mechanistic pathways in a variety of disease processes. Although the approach has proved powerful in identifying robust associations between many SNPs and traits, much additional work is needed to determine the functional basis for the observed associations so that appropriate interventions can be developed. Much more remains to be learned about how variations in intronic and intergenic regions, where the vast majority of SNP trait associations reside, influence gene expression, protein coding, and the disease phenotypes. Despite the limitations of using data obtained from the genome-wide association studies to assess the individual patient level of a risk for a particular condition, genome-wide scans may be useful in initiating counseling about non-genetic risk factors. Continued efforts to identify genetic variants that influence the response to drugs may yield new associations that could be used to tailor drug selection and dosing to the profile of the individual patient, particularly if it becomes possible to query these data through a user-friendly interface when a medication is ordered. Dear students, it was all about today's lecture regarding population and disease association studies. Hope you have understood it well. See you next time with a new topic. Till then, take care and bye-bye.